Welcome everybody on my lecture about HIV and AIDS. This is a continuation of chapter 3, immunopathy. And let me get to our PowerPoints so I can get going here. Okay, HIV and AIDS. This is a very nice rendering, artist's rendering of what the virus actually looks like. You can see all of these spike-like proteins on the surface of the capsid. The capsid itself is not circular, it's very geometric, and it's made up of a variety of triangles that are sort of glued together. So this is a pretty sturdy structure, and it certainly allows this virus to move from cell to cell as the infection spreads. So we have Nobel Prize winners again, two French researchers, and they won the prize for the discovery of the AIDS virus. Now, this was a very important discovery because the disease was actually ravaging throughout the world at this point. It was causing a pandemic, a global pandemic, and there were millions dying. So it was very important to know exactly what they were dealing with and the structure, and then how the structure was formed and the AIDS virus, how it replicated, and then use that information to develop drugs to destroy it. And that's pretty much what happened. We have some pretty effective drugs at this point to combat this disease. So AIDS itself is the disease. It stands for acquired immunodeficiency syndrome. The virus is known as HIV, and that stands for human immunodeficiency virus. So notice the word immunodeficiency. This tells you exactly what's going on here. When the virus is infecting somebody, it's actually destroying their immune system. And it does that by infecting the T helper cells. And this is their preferred target. So these are the CD4 cells that it's going after. The virus itself is an RNA retrovirus. That means that it will enter and leave the cell as RNA. And in order to replicate, it has to create its own DNA, its own viral DNA, that then moves into the nucleus and is inserted into the host's DNA and becomes part of it, becomes part of the genome, and then can replicate back into RNA from that DNA that it's created. Fantastic video here. This was produced by Khan Academy. This is one that you need to definitely watch. It's in your modules, and it basically explains how the virus is able to breach our barriers and then travel to our lymph nodes, where it will then infect those CD4 cells. So with the disease, what actually happens? Well, since the CD4 cells are the targets, their numbers will eventually decline, and then you start seeing the actual disease symptoms develop. And this includes the development of opportunistic infections. So think back to when we talked about a candidiasis, and you saw that slide on all of these white masses inside of this individual's mouth, on his tongue, on his cheeks. And at the time I said that that was going to be an AIDS patient, and that's because there is no resistance anymore to these particular infections, and they will just go rampant. Other things that can happen are a variety of carcinomas or cancers can occur. One is called Kaposi carcinoma, and in this particular case, you start seeing very diagnostic purplish splotches on the skin surface. You also get lymphomas. These are various blood cancers, and then other infections that can come up. And interestingly enough, TB is one that is critical here, and it is actually the leading cause of death from AIDS. And if you think about it, from what we learned about granulomas, if you remember, they're associated with TB and some other difficult to eradicate pathogens, but certainly with TB, you have those granulomas forming around the pathogen to contain them. And what part of those granulomas, an important part, are T cells and T helper cells. So when those T helper cells aren't around anymore, those granulomas will break down, and then the bacteria can just spread throughout the body and basically ravage the various organs. And that's something that you cannot recover from. So the course of the infection itself, this chart is a good one. It really shows what's going on. Here you have the initial infection. The darker green circles here are actually the levels of the CD4 cells. The lighter green circles represent levels of virus. So if we follow this along, and note that this goes, here we start with weeks of the initial infection, and then we go through years, and we actually get to year 11 and 12. So this is a very kind of long developing type of disease till you get to the end stage. But what happens initially is that you start out with a pretty good level here of your CD4 cells. Here you get the viral infection starting. Initially, the infection takes off and lots of virus. You see a lot of symptoms. CD4 cell drops. They're able to recover though. They don't get back to those higher levels, but initially they recover and are able to control the virus. You see how the viral loads drop. And even though the CD4 cell numbers are decreasing, they're still able to keep this virus in check until a critical point is reached. And that's about in this area here, about 7-8 years. And then you just reach a point where those CD4 cells can't provide the immunity anymore needed to contain the virus. The virus loads again shoot up. You start seeing symptoms again. You get opportunistic diseases. Eventually, those CD4 numbers crash and viral loads are high, and you have death. So how do these viruses replicate? And that's quite interesting, actually. HIV is actually an RNA retrovirus, which means that it enters a cell as RNA and it leaves the cell as RNA. In order to replicate, it brings along a variety of enzymes that help it in the process. So you have reverse transcriptase. So this is what creates that viral DNA from the viral RNA. You have integrase. So this allows the adding in of the viral DNA into the human DNA. And then finally, you have proteases, and these are actually enzymes that process the viral proteins so that the enzymes are made, the enzymes that it needs to take with it to its next cell are made, along with the capsule, along with everything else, the spikes, in order with everything so that it can infect other cells. So AIDS treatment. Fortunately, the medications for AIDS are actually very effective at this point, and they primarily target the HIV replication. So there are several antivirals presently in use. We have some that will inhibit the entry of the virus into the cell. There are others that prevent the actual replication, so the creation of or the ability of the reverse transcriptase to be effective. There are others that prevent the newly formed DNA from actually integrating into the host DNA. And then finally, you have protease inhibitors. So these are the ones that will inhibit the creation of the capsid or the spikes or whatever's protein here, new enzymes, all the things that are needed for that AIDS virus to move on. So what was discovered was that just taking one of these is not going to be effective enough. So they have developed what are called medication cocktails, where they combine different ones of these into one dose that's then taken. So by hitting the virus in a number of different places during this replication process, they're able to get very effective treatment. And what we see is that that the patient becomes asymptomatic, returns back to life, a normal life, but is AIDS actually cured? Not yet. The disease is still dormant and it can reoccur. So it's chronic. But as long as the medications are effective, of course, they will halt and reverse any infections that crop up. The important thing is that the medications are taken daily. And by doing that, those viral loads that I was talking about, they become undetectable, and the CD4 cell counts are actually able to return to normal. So the good news is that long-term remission is possible, and many of these patients die from completely unrelated AIDS problems. The downside of all this, of course, is that these medications themselves are pretty toxic. They're very effective, but they can have negative effects on the patient. And occasionally the effects can be fatal, so the person can die from that. Or the virus, which mutates at an incredibly high rate, just becomes a form that the medications can't handle anymore. And to end, there's a great video here that sort of summarizes everything that I mentioned. This is something that you do need to watch. It'll reinforce everything that I have gone over. And this is something that I will be testing you on. So be sure to watch the videos, review these notes here, these PowerPoints, and make sure that you understand the material. Okay, class, I will see you next week for our next lecture and actually our midterm. So study and just be prepared. See you next time.